In our final lecture on muscle physiology, we are going to look at some molecular mechanisms that can lead to the symptoms of fatigue, and then we'll briefly look at changes in muscle mass. In addition, there's something I should have covered in the previous lecture but didn't, and so I'll cover it right now. In the previous lecture, we looked at fuel sources as it applied to exercise intensity. So when the exercise is a very high intensity, like 95% of your maximum heart rate, then you're predominantly utilizing glucose anaerobically to replenish your ATP. But when your exercise intensity is low, you predominantly use aerobic metabolism, and fatty acids are preferred. So where is the location of glucose and fatty acids? Well, at rest, your skeletal muscle will actually stockpile glucose in the form of glycogen and fatty acids, but it's not an unlimited supply. So as that muscle starts contracting during exercise, you'll start to burn through your supply of fuel. Much like filling up your car with gas, it's not unlimited. As you start driving, the fuel drops. And at some point, you're going to have to refuel. So where do you get additional fuel from? Your blood. So glucose and fatty acids move around the blood and then can get drawn into muscles that are in use or exercising muscles. So if you're exercising pretty strenuously, 75% of max heart rate or greater, the amount that is stored in your cells will last 20 minutes or so. And then after that point, your fuel reserves will get low and you'll start pulling it in from the blood. By 60 minutes into your exercise, let's say you're running a half marathon, you're going to be pulling mostly from the blood. So I'm a terrible runner, but I do train, kind of, sort of, for half marathons. It's really kind of stupid. Like I said, I'm a really terrible runner. It's the hardest thing for me to do, so I've decided that I'm going to do that because I'm really stubborn and don't like to be told I can't do something. So that's a personal issue of mine, but okay. So I obviously train for half marathons, and one of the things that I found is that, by like 7 or 8 miles in, I would just be completely gassed unless I ate. And so I started eating during my runs, and eating, of course, brings more glucose in. The glucose goes into my blood, and lo and behold, I could run a lot longer. So some people run whole half marathons without eating anything, and I honestly do not know how they do that. It's not as if your recent meal is your only source of glucose and fatty acids. You can liquidate fatty acids from your adipose cells, and you can liquidate glucose from your liver. But bringing it in from food is a pretty easy way to get it into your blood. So it works for me. It doesn't work for everybody, but it does underscore the importance of refueling your cells during exercise. So with that note, let's move on to fatigue. So we're going to start by defining fatigue. Now, I want you to envision that I have an animal muscle fiber, a real one, a living one, and I've stretched it out and I'm holding it, and I've got some sort of equipment that I can measure how much tension is generated in a twitch. So I can actually take an electrical wire and kind of poke at it, create a localized depolarization which will radiate out and cause a twitch. So I'm pulling the skeletal muscle fiber and I say poke and it goes dunk, twitch. And poke again, twitch. Poke again, twitch. Poke again, twitch. And I keep doing that over and over and over and over again. And at some point, what I'm going to see is that the amount of tension that is generated from a twitch that's being measured by my machine is going to drop. So in other words, the amount of tension generated in some of the later twitches is going to be less than the amount of tension generated initially. And nothing else about my system has changed, other than my skeletal muscle has fatigued. So fatigue is defined as a skeletal muscle. It can be a fiber, it can be the whole skeletal muscle, that is not generating the amount of tension that it should generate. It's less than that because of prior use or prior twitches. So something about earlier twitches is causing it to tire out and fatigue. So in lab, we actually get to see this happen, where somebody takes a hand dynamometer and squeezes it as hard as they possibly can. And they actually close their eyes and they squeeze and they squeeze and they squeeze and they think that they're maintaining the same tension. But on the computer screen, you can see the tension drops, and that's fatigue. And it sets in within seconds. So it's not like we're talking 3 hours later. We're talking like seconds later. Some types of muscle fibers fatigue faster than others. And as you might expect, type 2X or fast twitch glycolytic fibers fatigue within seconds, whereas slow twitch fibers can actually go for hours. So the converse of fatigue is endurance. So when we think about skeletal muscle fibers with high endurance, that means that it takes them a long time to fatigue, whereas skeletal muscle fibers with low endurance fatigue very quickly. Okay, let's go ahead and talk about some of the potential mechanisms that underlie fatigue. In other words, why does fatigue happen? What is it about prior use that is causing a loss of tension? And a long time ago, people thought that the skeletal muscle fibers ran out of ATP. And we've since thoroughly rebuked that idea. It's not that the skeletal muscle fibers run out of ATP. Thank goodness, because then you'd be looking at serious muscle damage. Fatigue does not have one cause. There are probably a dozen molecular things that are all going on that contribute to the loss of tension. But before we start getting into some of the potential causes, I want to remind you that tension is directly proportional to the number of cross bridges. So if I'm really talking about fatigue, then what I'm really saying is that I'm not generating as many cross bridges as I think I should be. Interesting idea. Okay. So what are some potential reasons for that? In this lecture, I am going to cover 3 potential mechanisms. 2 of them are pretty well established, and one of them is a little bit hypothetical. And this is not an exhaustive list. Again, there are many known causes or contributors to the symptoms of fatigue. There's not just one cause. The first one is pretty well established, and I would say the most well known among the general population. And that is the buildup of lactic acid that comes from the anaerobic metabolism of glucose. So it would make sense then that we see this most often with type 2X glycolytic fibers. Okay, so why does this cause problems? Lactic acid is an acid, and because it is an acid, it contributes a hydrogen ion to the cytosol. So when lactic acid starts building up, the pH of the cytosol starts dropping. Well, this interferes with protein folding. And some of the proteins that are potentially most affected by this are myosin. So myosin heads seem to maybe partially denature and don't work as well. And if they don't work as well, they're not going to generate as many cross bridges, and therefore you're going to get the lack of tension. So I think that one probably is also the most clear cut. All right, here's another thing that is actually pretty well established, though the mechanism of why this happens is not 100% clear. So there is evidence that in a fatigued muscle, the sodium-potassium pump only works at 30% efficiency. That's pretty strange. I'm not exactly sure why that's true, but it is observed. So they've done studies and they've shown that, hey, in a really fatigued muscle, the sodium-potassium pump doesn't do its job very well. It works, let's just say, 30% of the time. Okay, why is that critical? Well, let's consider what we expect to happen during a twitch. We know that in excitation-contraction coupling, we're going to start with the motor neuron firing action potentials down the axon terminal. This is going to lead to secretion of acetylcholine that's then going to bind to, put that here, a nicotinic receptor that's going to open up the nicotinic gate, and guess what? Well, sodium needs to come in. And that works as long as there is a really strong chemical gradient where sodium desperately wants to come in. But remember that another thing that happens is we have action potentials propagating across the sarcolemma, right? And so when those action potentials propagate down the sarcolemma, some of the sodium moves in and some of the potassium moves out. So let's just assume for a moment that the sodium-potassium pump takes a long lunch break and decides not to do anything for a while. But we keep getting action potentials. So sodium keeps coming in, potassium keeps coming out, sodium keeps coming in, potassium keeps coming out. And so over time, the gradients are not nearly as strong. So now this cell doesn't have a very strong gradient of either sodium or potassium, and this motor neuron says, okay, time to go again, and you fire action potentials, we release acetylcholine, we open up this gate, and does any sodium come in? Well, if it does, it's probably not enough to create the graded potential needed. So in other words, no action potentials happen. If no action potentials happen, there's no calcium released. If there's no calcium released, then there's no calcium binding to troponin. And if there's no calcium binding to troponin, cross bridges are not happening. So this is kind of an extreme example of what would happen if the sodium-potassium pump just completely stopped working. There would be no action potentials, and therefore there would be no cross bridges. You would have no tension, zip zero. But if it's only working at 30% efficiency, it is trying to reestablish the gradients, but it's just taking its sweet time. And it's not doing it very fast. So let's assume for a moment that the gradient currently looks like what you see on the screen. And this muscle fiber fires and we try to start an action potential. But that gradient is just not strong enough to let enough sodium come in to start an action potential. So no action potential happens. Okay, well, you know what? This motor neuron is actually trying to summate twitches, so it fires again. But meanwhile, I've worked a little bit and tried to restore a little bit more of the gradient. And now one of these comes in, but you know what? That's just still not enough depolarization. So no action potential happen. But I'm going to keep working a little bit more, again, working real slow here because I'm on 30% efficiency, and I open up this gate again, and now there's enough of a gradient for two of these guys to come in, and now we've got action potentials. Okay, So in other words, this motor neuron fired 3 times, but the muscle only responded once. In other words, only 30% of the motor neurons' firings actually resulted in a release of calcium. So you could see then how you could never achieve tetanus under this scenario, and your twitches would poorly summate. What your body would want to do is something like this, all the way up to tetanus, and what it would be doing is something like this. And so therefore, those are the symptoms of fatigue. So again, less frequent action potentials, certainly not matching what the motor neuron is dictating, less frequent release of calcium, less cross bridges. All right, so the third one is kind of more of a hypothesis. I honestly do not know if there's experimental data to support it, but it's certainly been postulated that this could be happening. So one of the things that contributes to cross bridges, of course, is the exposure of actin binding spots. And in order to do that, calcium, which I have here as pink dots, needs to bind to troponin, and then that moves the tropomyosin out of the way. Well, the reason why calcium is attracted to troponin is because troponin probably has a negative charge in there. And that attracts the calcium. But in a muscle that's been doing a lot of cross bridges, the myosin has also been spitting out ADP and phosphate groups. And those phosphate groups are also very close to this tropomyosin and troponin. So when calcium is released from the sarcoplasmic reticulum, you think to yourself, oh, it should go here. But maybe it kind of spends a little bit of time hanging around with another negatively charged ion, because positively charged calcium is going to be attracted to pretty much anything negatively charged, right? So if that's the case, then phosphate kind of distracts calcium. And that means there's not as much calcium binding to troponin, and therefore there's not as many actin spots exposed for myosin to grab onto. So in this example, what is postulated is that a heavily worked muscle is going to have an excess or an abundance, and a very local abundance, of phosphate groups. And phosphate groups by themselves have a -3 charge to them. And this is again because of the cross-bridge cycle. The myosin head spits out an ADP and a phosphate group as part of that cross-bridge cycle. Now eventually ADP and that phosphate group are going to rejoin and make ATP, but initially there's just a lot of phosphate hanging around. And maybe it distracts some of the calcium and prevents calcium from binding to troponin, and therefore more of the binding spots remain covered than should. And that could be another contributor to fatigue. So these are 3 potential contributions to fatigue, 2 of which are pretty well established. The third one is still kind of just hypothetical. I don't know that there's a lot of data to support it, but it was postulated, and it kind of makes sense that that might happen. Our last topic is going to be changes in muscle mass. So when you have increase in muscle mass, we have a name for that. We call it hypertrophy. And when we have a decrease in muscle mass, we call it atrophy. Muscle mass changes are very common. Muscle is incredibly responsive to your daily activities and use. So my question here is, how does a muscle hypertrophy or how does a muscle atrophy? And I'm really just looking for a 1-sentence answer here. So let me tell you what it's not. The number of actual muscle fibers does not change significantly either way. And you can kind of see that here. When you look at the difference, the number of muscle fibers doesn't look particularly different, but rather the diameter of the muscle fibers looks different. So when You have a skeletal muscle fiber, like so. During hypertrophy, the individual myofibrils that are inside here are going to get bigger. And what that's going to do is that's going to take the individual muscle fiber, and I'm exaggerating here, but it's going to make it much bigger. So the individual myofibrils are getting bigger. So hypertrophy is achieved by making individual fibers bigger, and that's done by either enlarging myofibrils within the fiber or potentially having more myofibrils. But you don't add additional fibers, so the number of fibers is relatively unchanged. And atrophy is exactly the opposite. Under atrophy, the fibers get really small, the myofibrils get really small, and therefore the entire fiber shrinks. So again, it's not usually a reduction in number of cells. So if I ask you on the exam, true, false, muscle hypertrophy is achieved by adding additional muscle fibers, that is a false statement. So the main way that muscles hypertrophy is simply by enlarging existing fibers. Now you can add more myofibrils, but you're not going to be adding more fibers. Again, there's some exceptions to this, but this is kind of the general trend. And with that, we end our lecture series on muscle physiology.